Welcome back, everyone. Thank you so much for joining us for the second session. Before we start, I would again love to remind you that you have a chance to ask questions for exciting panel session that's waiting for us at the end of the day. So please don't forget, share your questions with us. And now I would like to welcome our first speaker, Dr. Ellery from Monash University, Australia.
Hi, my name is Stacey Ellery and I'm a medical researcher at the Hudson Institute of Medical Research and Monash Universities in Melbourne, Australia. It is my great pleasure to present this presentation to you on creatine metabolism in female reproduction, pregnancy and newborn health. So female reproductive organs are some of the most regenerative and highly energetic tissues within our body. And so it's not surprising that there is an undeniable link between adequate energy metabolism and successful reproduction. Now, given what we know about the importance of creatine for energy homeostasis in tissues that experience either fluctuations in energy or high energy demands, it is not surprising that a few questions come to mind, particularly whether creatine metabolism might be fundamental in female reproductive tissues, the developing fetus and the neonate. Given that we know that 50% of our creatine comes from our diet and that creatine levels would likely go up through reproductive processes, a secondary question that comes to mind might be whether dietary creatine is conditionally essential during pregnancy. And then finally, whether we could use maternal dietary creatine supplements during pregnancy to help buffer changes in energy demand associated with common pregnancy complications. Because an underlying feature of a lot of pregnancy complications is poor oxygen delivery or hypoxia-ischemia to either the placenta or to the fetus. So if we look at creatine in the female reproductive literature to date, I liken it a little bit to a Where's Waldo. Creatine has been popping up in the literature for the last 60 years, but it's been somewhat sporadic and I guess a little ad hoc. So there's still no real consensus on how important creatine metabolism is for successful reproduction. So to help try and push this field further and consolidate what we do know so that we know where to take the research as we move forward, our team put together this extensive review on creatine metabolism in female reproductive health last year. And it is these topics that I will be covering today in some detail. Now, because of time constraints and a desire to show you some new and exciting unpublished data, I'll just be focusing on a few aspects of creatine metabolism in female reproduction. But if anyone has any questions about topics that I don't cover today, you are more than welcome to get in contact with me. So first, let's look at the female reproductive system. We're going to focus today on the uterus and the two key layers of the uterus, that being the endometrial layer, which is the layer that is cyclically shed through the menstrual cycle and rebuilds itself every month, and is very important for embryo implantation and establishing pregnancy. And we're also going to talk about the myometrium, which is the smooth muscle layer of the uterus, which is very important for labor progression and contractions. So firstly, to the endometrium, we know that creatine can be and has been detected in the human endometrium, and it is particularly present during the secretory phase of the menstrual cycle. Now, this is important because it's the secretory phase where any fertilized embryo would be seeking to implant. So this is a highly energetic period of the uterine cycle. And it's not surprising that along with that higher creatine level at that secretory phase of the menstrual cycle, there's also an upregulation in expression of the creatine transporter to help perhaps facilitate that uptake of creatine, and also an upregulation in the creatine kinase CKBB enzyme of the creatine circuit. So that enzyme, which is essential for actually helping generate ATP from stored phosphocreatine. Now, whether or not these changes are associated with pregnancy outcomes or the success of pregnancy is still undetermined, but there is some exciting data that's just come out of Rod Snow's laboratory at Deakin University here in Melbourne. So what Rod did with one of his PhD students, Mametha Phillip, was look at the level of the creatine synthesizing enzymes, CKBB, and the creatine transporter in endometrial sections from women collected at different stages of the menstrual cycle. He also looked at the level of expression in women who were considered fertile and also those with primary infertility, so women who were struggling to fall pregnant. And I just want to focus our attention on the creatine transporter again. We can see that there is this nice increase in creatine transporter expression across the early phases of the menstrual cycle, peaking at that mid-secretory phase. So similar to the previous data, we can see that the expression level is going up around that time when a fertilized embryo would be seeking to implant into the endometrium. But we can see that there isn't actually that increase in creatine transporter expression present in those women experiencing primary infertility. So this requires a lot more work, but it's certainly a clue to whether or not creatine metabolism might actually be contributing to conditions, or dysregulation, I should say, of creatine metabolism might actually be contributing to early pregnancy loss conditions such as failure to progress or early miscarriage. Now moving on to the myometrium, so the muscle layer of the uterus, we can see it's not surprising that phosphocreatine has been detected in high levels of this muscle layer at the end of pregnancy compared to a non-pregnant tissue. So there appears to be a buildup of phosphocreatine in preparation for labour. We also see that CKBB enzyme is detected in that layer of the myometrium, so there's likely the use of the circuit to actually utilise that phosphocreatine for ATP production, and that again there is a threefold increase in the expression of that enzyme at term compared to early gestation. And then finally, I just wanted to point out that again, new data which will be published out of Professor Rod Snow's lab later in the year, he and his PhD student have just shown that along with the presence of creatine, phosphocreatine, and the creatine kinase circuit enzymes, that the myometrial layer of the human uterus at term also expresses the AGAT and GAMT enzymes required for creatine synthesis. So it may be that this tissue can actually produce its own creatine to help build up those stores prior to labour. So now moving on to the placenta. So once you have an established pregnancy, you obviously have development of the fetus, but you also have simultaneous development of the placenta. And so the placenta is a wonderful organ which effectively takes on all of the responsibilities and all of the organ functions of the fetus while it is developing in utero. And so uniquely so, it is an organ that needs to actually grow and develop whilst also being functional. So again, a highly metabolic organ, consumes between 40 to 60% of oxygen and glucose, which is supplied to the uterus during pregnancy. So given the bioenergetics of the placenta and its high energy demand, it's not surprising that creatine and phosphocreatine have been detected in the human placenta. Most of these studies have been done at term, but it is likely there from early on in gestation. There was also a very nice study by Thomure, who published in 1996, which showed expression of the creatine kinases in all 3 trimesters of human pregnancy, and that the expression pattern of the creatine kinase enzymes reflected the likely energy demands of the placenta during that time. So effectively, the level of enzyme expression increased up until the end of the 2nd trimester, where energy demands hit their peak, and then started to tail off towards delivery, which is what we see happening in placental energy metabolism as a whole. And so with this known, we were then curious a couple of years ago as to whether or not the human placenta could actually synthesize its own creatine and where the expression of the creatine transporter might be. So which cells within the placenta might actually be best utilizing creatine for energy homeostasis. And so we conducted a simple study where we took 12 healthy placental samples at term pregnancy. These were non-labouring samples, and looked for the expression of the creatine synthesizing enzymes and the creatine transporter. And indeed, we found that the human placenta did express the two enzymes required for creatine synthesis, AGAT and GAMT, and also the creatine transporter. And we found that the creatine transporter expression was on some specialized cells called the syncytial trophoblast, which is basically the cell structure which sits at the interface between the maternal and the fetal circulations. So these cells are considered as the most highly energetic cells within the human placenta. So it's not surprising that it is likely this cell population which is utilizing creatine for ATP homeostasis. So given that the human placenta could produce its own creatine, and there was evidence that creatine might be really important for placental bioenergetics, we were then interested to see whether placental creatine supply and utilization might actually be altered with common placental pathologies, because most common placental pathologies are associated with reduced blood flow to the placenta, thus reduced oxygen and glucose supply and energy deficiencies. And so I won't lie, I got really excited when we started these studies because I thought, or I hypothesized that a placenta which was compromised would actually have lower creatine levels because there wouldn't be that capacity to synthesize creatine as well as you would under normal or healthy circumstances. So delivery of those key amino acids, the required ATP production in the first place, I hypothesized that this would all have declined in pregnancy complications associated with placental insufficiency, and then got excited about the idea of perhaps using a dietary creatine supplement to help boost those creatine levels and the overall function of the placenta. But alas, like what happens in science quite often, I was incorrect with my hypothesis. So I'll just share with you some data that has come from two placentas collected from two pregnancy complications, those being fetal growth restriction due to placental insufficiency and also preeclampsia. And what we saw quite clearly when we looked at the creatine content of placentae collected at delivery from pregnancies experiencing these complications was a significant increase in creatine levels within the tissue. So placentae collected from pregnancies complicated with fetal growth restriction had 43% more creatine and, interestingly, 37% less GAA, so that precursor to creatine that is produced during that first stage of creatine synthesis. So more creatine, less creatine precursor GAA compared to healthy controls in a fetal growth restricted pregnancy. And we also saw that in placentae from preeclamptic pregnancies that the creatine content was 38% higher than in healthy controls. So there seemed to be a natural adaptation in these pregnancies that were experiencing energy failure and placental dysfunction to actually increase creatine levels within the placental tissue. Now, the mechanisms behind this are still not very well understood and something that we'll be looking at a little bit further with our studies, but certainly an interesting finding that there seems to be this natural adaptation in this pregnancy tissue to increase creatine when there's suboptimal environment which will compromise overall energy metabolism. So given that the placenta requires creatine, we know that the fetus requires creatine, and that those creatine levels would likely need to increase with advancing gestation as the placenta and the fetus grow and the myometrium obviously prepares itself for delivery. We then wanted to consider where this provision of creatine is coming from. Are there adaptations to maternal creatine metabolism during pregnancy which are facilitating these changes? And so to start to address these questions, we have just recently completed our Creatine in Pregnant and Pregnancy Outcomes, or CPO, Prospective cohort study. And so this was a cohort study where we saw around 280 healthy women across their pregnancies coming through our system at Monash Health to collect biological samples and nutritional data, as well as pregnancy outcomes data, so that we could start to tease apart what was happening in terms of their creatine metabolism. And at this point, I'd just like to say a huge shout out to Deb de Guingand, who conducted a lot of this work throughout her PhD. She was a research midwife who joined our team and was absolutely phenomenal on this project. And also Dr. Kirsten Palmer, who is a clinician scientist at Monash Health and maternal fetal medicine expert. So they were absolutely fundamental in the completion of this study. And so the protocol for this study has been published if you'd like to have a look at it in detail, but effectively we tried to see our participants 5 times throughout their pregnancy when they were coming into the hospital to receive their antenatal care. When they came in, we took a blood and a urine sample and also a 24-hour food recall. Then when the women came into birth, we collected a sample from their placenta. We took a sample of mum's blood, a urine sample from mum, and also collected samples from the 2 circulations of the placenta. So we have a venous cord plasma sample, which is effectively the blood flowing from the placenta to the fetus, and also an arterial cord serum sample. So that is the blood that is circulating around the fetus and then being returned to the placenta. And so we hopefully will be publishing this data in a series of papers in the next coming months. It was an incredibly large study, so at this stage we're looking at publishing it across 3 manuscripts, so keep an eye out for those. But just some of the key findings that we found in terms of overall creatine homeostasis during pregnancy. I'll present to you some data on creatine itself, but also on GAA, so that creatine precursor produced during endogenous synthesis, just to give us a little bit of an understanding of how de novo synthesis might be shifting to help support creatine levels across pregnancy. So firstly, we saw that within our 280 odd women, from which we had just over 1,200 samples across pregnancy in which to measure creatine, that the maternal plasma creatine concentrations remained remarkably steady throughout gestation, sitting around the 36 to 450 micromolar range. So not too dissimilar to what we see in a non-pregnant population. So this is despite likely draws on maternal creatine to support placental creatine accruement, the fetal creatine requirements, and also changes to maternal mass adaptation and physiology during pregnancy. So despite all of these changes and a likely need for more creatine, maternal plasma levels stayed very consistent. However, maternal urinary creatine excretion declined with advancing gestation. So it was likely that there might be a drawback on excess creatine towards the end of gestation when those creatine requirements peak to maintain that steady state in the maternal plasma. And again, when we looked at the GAA concentrations, that theory tended to hold true as well, where we saw that maternal plasma GAA levels, unlike creatine, did fluctuate quite a bit across pregnancy, with an overall falling concentration as pregnancy advanced. The opposite was seen with maternal urinary GAA. So as pregnancy advanced, we saw a decline in, a rise, sorry, in maternal GAA excretion. So effectively, this data suggests that maternal creatine levels are maintained steady, but there are changes in creatine excretion and perhaps GAA production and GAA excretion, which are helping to ensure that that steady state is maintained. Now, when we look at creatine concentrations at birth, interestingly, we found that creatine concentrations in the venous cord plasma declined with advancing gestation. So there's perhaps a limit to how well this system can be maintained as pregnancy is advancing. And that at the same, with advancing gestation, placental GAA levels started to decline. So again, this system might be being pushed to the limits towards the end of pregnancy. And that we also saw, which was interesting, that placental and arterial cord serum creatine levels were actually significantly higher at birth in pregnancies which were carrying a male fetus compared to a female fetus, which is quite an interesting finding. We know that the way in which the male and female placenta, their metabolism is actually slightly different, and the way in which growth of the placenta and fetus as a ratio is matched is slightly different as well, which means that male fetuses tend to not do as well during pregnancy complications associated with either hypoxia or other disturbances to energy metabolism. So it was interesting to see that in the male fetuses, they seem to have higher creatine levels sitting there ready to go. Now, in terms of creatine on neonatal growth parameters, I think this is a really important point to make, and that is that overall, in this low-risk pregnancy cohort of around 280 women, we found that maternal, cord blood, and placental creatine concentrations had no impact on birth weight, birth length, or head circumference. So I guess that was not overly surprising considering how steady and stable creatine was throughout gestation, but an important finding and a new one at that. So something to definitely stress as we talk about some of the other characteristics of creatine metabolism in this particular cohort. So whilst creatine didn't have any associations, GAA did. So we found that concentrations of that creatine precursor GAA, which is produced during de novo synthesis, was associated with changes in birth length and head circumference. They were modest changes, but effectively the higher the GAA levels, the baby pregnancies with higher GAA levels had slightly longer babies with slightly bigger heads. And so then let's look at whether maternal characteristics were actually modulating creatine availability. And we were actually quite surprised to see that there were a lot of maternal characteristics that were adjusting creatine levels. So they included age, parity, ethnicity, BMI, smoking status prior to pregnancy, anemia, vitamin D deficiency, and hypothyroidism. Now, unfortunately, due to time, I don't have the capacity to go through the ups and downs in detail here. They'll obviously come out in the papers in the next couple of months. But effectively, in these conditions that are associated with slightly poorer energy metabolism or changes in oxygen delivery and oxygen metabolism, we saw that there was overall a decrease in creatine levels, but a compensatory increase in GAA. So again, this requirement or this likely adaptation to try and increase creatine levels through upregulating de novo creatine synthesis. One maternal characteristics that I would like to focus on slightly today, because I'm sure it is of interest to a lot of people in the audience, is actually maternal diet, and a diet which is devoid of animal protein. So we know that if we do consume animal protein through our diet, that around 50% of our creatine comes from that source. And so we're obviously curious as to whether that holds true in pregnancy, and whether women who elect to not consume animal protein during their diet, whether that actually alters their creatine levels throughout pregnancy. And so because with this cohort, when we collected a blood and urine sample throughout gestation, we also collected a 24-hour food recall, we were actually able to do a direct association between the amount of animal protein intake taken in the 24 hours prior to a blood sample and the amount of circulating creatine. And we found that at every stage of pregnancy, that there was indeed a significant association between the two. So overall, for every gram increase in animal protein intake in the 24 hours prior to the blood sampling led to an increase in plasma circulating creatine of between 0.07 and 0.18 micromolar, depending on the stage of pregnancy. And so with this in mind, we were then interested to look at the levels of creatine in our non-meat eaters. And so we did a nested study within the CPO cohort to look at non-meat eaters and meat eaters, as correcting for any other maternal characteristics or associations that might be modulating creatine levels. And indeed, you can see from these graphs over here, with our non-meat eaters in blue and our meat eaters in red, that our meat eaters had substantially and significantly higher circulating creatine levels throughout pregnancy. And that difference between the two was around a 30% increase, or a 30% reduction, if you want to think about it the other way, in our participants who were not consuming animal protein through their diet. And we can see that their urinary creatine excretion was following a similar pattern. Again, interestingly, there is some evidence that the body might be trying to compensate for this decline in creatine availability through the diet through upregulating de novo creatine synthesis, as we saw the opposite effect with plasma GAA levels. So we can see here in the blue that our non-meat eaters had higher plasma GAA levels across pregnancy compared to our meat eaters, and that whilst not significant, the pattern of urinary GAA excretion was also the opposite between the two populations. Now, again, I just want to stress that in this nested cohort study, where we looked at around 30 participants in each group, we found no difference in outcomes for these pregnancies. So the difference in creatine levels in our non-meat eaters had no direct effect on neonatal outcomes. And this is supported by epidemiological data. We know that selecting to have a vegetarian or vegan diet doesn't necessarily directly link to poorer pregnancy outcomes. But where we think this might be an interesting finding and something that should be communicated with women who choose to have a diet devoid of animal protein is that we do see some interesting associations between creatine and labour outcomes. So what we found in this study was that depending on the way in which you delivered your baby, the amount of creatine in the venous, in the cord bloods actually changed. And so the take-home message from this is that basically the more energetic and energy-intensive forms of labour, so those being a vaginal unassisted delivery or a vaginal instrumented delivery, including an emergency C-section, these all led to there being higher levels of creatine in the venous cord plasma after delivery. So this is the blood supply which is going from the placenta to the fetus. So during these forms of delivery where the fetus can become distressed at times, or the fetus already has become distressed and they've elected to intervene, we see that there's higher creatine delivery to the fetus. And so Again, so that was with the venous cord levels, and this was also reflected in the arterial cord serum samples as well. So the actual blood sample, the blood that is circulating around the fetus. So there seems to be this adaptation during labor to get more creatine to the fetus, particularly during energetically active labors. And so again, whilst I stress that the lower creatine levels in the women who were not eating meat were not adversely affecting their babies, because there seems to be this natural adaptation in high energy scenarios to get more creatine to the fetus, it is interesting to think about whether that reduced creatine availability in the first place might actually in any way compromise this natural adaptation. And then the last point I'd like to make here is that we also, and it just furthers this point, when we looked at the correlations between the plasma creatine levels at the end of pregnancy with the first stage of labor. So the longer that women labored for, the higher the levels of creatine in the venous cord plasma that were detected after delivery. And conversely, we saw that after the longer labors, that venous cord plasma GAA levels declined. So again, that potential there for there to be trying to be a rebalancing of the creatine being delivered to the fetus to ensure that there's optimal creatine being delivered. And again, this was just reflected too when we looked at the correlation between the venous cord and arterial cord serum creatine concentrations and cord lactates. So lactates are taken at the end of labor as a measure of fetal distress and how hypoxic, I guess, the fetus became during the labor or the delivery process. And again, those fetuses with the higher lactates, so those that experience more distress, had higher circulating levels of creatine. So finally, I just wanted to touch on fetal creatine metabolism and the use of creatine supplements for the fetus. And so obviously we know from the wonderful work of Olivier Bressant and his team over quite a long time, it is clear that particularly for the fetal brain, that creatine metabolism is really important from early in the embryonic period all the way through until delivery. And so considering that one of the most severe complications of some pregnancy issues tends to be perinatal brain injury, we were interested in whether a creatine supplement could be used to increase creatine levels in the brain, fetal brain, prior to an insult. Particularly, we've looked at hypoxic insults. And so with that higher phosphocreatine being on board prior to the oxygen deprivation, whether you would have a better chance of maintaining ATP production and thus reducing perinatal brain injury. And so this slide here is just to point out from a mechanistic perspective, the rationale behind why creatine might be helpful in this space. And really, we would be focusing and have been focusing on two pregnancy complications, that being preterm delivery of the baby and also intrapartum asphyxia. So intrapartum asphyxia being a severe acute hypoxic insult to the fetus during labor. So this work was really started by Professor David Walker in our team about 10 years ago with the wonderful spiny mouse. And just in quick summary of that, he found that if we supplemented our pregnant mice with creatine for around 20 days, we increased pup survival after an acute hypoxic insult at birth. This creatine protected against brain pathologies. It also seemed to protect the neonatal kidney from injury both in the neonatal period and later in life, and was also able to protect some structural changes to the offspring's diaphragm and skeletal muscle. So certainly some clear indications that maternal dietary creatine supplementation may be beneficial to protect multiple fetal organs from a hypoxic insult at birth. And what I'd like to share with you today is some further work that we've been doing in this space using a fetal sheep model of acute hypoxia. And this has really been undertaken by two previous PhD students in our laboratory who have just recently received their doctorates. So Dr. Ni Tran and Dr. Anna Muccini, who did a fetal supplement of creatine in utero prior to doing an umbilical cord occlusion to induce a hypoxic event in the fetus, and then focused on some of brain metabolism and some of those outcomes. So what we can see from some of Ni's work, which is incredibly exciting, she measured reactive oxygen species accumulation in the fetal brain after the hypoxic insult in real time, and found that overall the level of ROS production in the creatine supplemented animals was decreased compared to the controls. And this was particularly in the first 24 hours after the hypoxic insult, which is when we know that the injury cascade, which produces perinatal brain injury, is manifesting. So a very, very exciting new finding, which will hopefully be published in the next couple of weeks. This data is currently under secondary review. Now, in terms of what that change is doing to overall the health of the fetus, we have seen some interesting results actually. And so one that I just wanted to point out was that in our saline UCO animals, if we looked at the creatine content in the striatum region of the brain 72 hours after the hypoxia, we actually found that the creatine concentrations were similar to those of the supplemented animals. So again, following on from the human data, there's this nice pattern of a natural adaptation of getting more creatine either to the placenta or to the fetus during energy collapse or periods of high energy demand or hypoxia. So this is an incredibly interesting development in this space, that there is this natural adaptation to get creatine to these tissues which are under distress. And so in the context of our fetal sheep models, what this overall means that has led to is that our fetuses with the higher creatine levels had lower systemic lactates, higher partial pressures of O2 and oxygen saturations after the UCO, which overall resulted in reduced increases in cerebral lactate, glutamate, and pyruvate in the brain. Now, in terms of whether that has any functional consequences, we're still teasing that out. But again, some wonderful new data, which has come from Ni in a collaboration with Dr. Robert Galinsky at the Hudson Institute, where they monitored fetal EEG or brain activity after the hypoxia in real time. They found that our creatine supplemented sheep who had experienced a moderate hypoxia actually had reduced seizure burden in the 72 odd hours after the hypoxic insult. So there was a significant decline in total seizure duration, total seizure number, and overall seizure burden, which again, this is an incredibly exciting new finding and provides further evidence of the utility of using creatine supplements in pregnancy to protect the perinatal brain. So just to conclude with a few take-home messages, going all the way back to the start of focusing on the female reproductive system and cyclical changes in the uterus, which are required to support the actual development of pregnancy, we can see that there is clear evidence that both the endometrium and the myometrium can use the creatine kinase circuit for energy homeostasis. And there are starting to be some links between endometrial creatine metabolism, particularly in the stage of the menstrual cycle where there is embryo implantation, and shifts in creatine availability could potentially be linked to primary infertility. So an incredibly interesting area which needs to be explored a little bit further. Because we should be perhaps considering whether creatine supplements would be beneficial to help support that phase of uterine metabolism and implantation. And that could be in both the space of natural pregnancies, but also in the space of artificial reproductive therapies, or those couples or individuals undergoing IVF. In terms of creatine metabolism and shifts during pregnancy, there are certainly shifts in maternal creatine metabolism throughout pregnancy, but really they seem to be shifts which are in place to try and keep maternal creatine levels actually stable for the duration of the pregnancy. We've seen that there are a number of maternal characteristics and maternal conditions that are monitored during pregnancy, which are actually modulating creatine metabolism. And that changes in GAAs or shifts in endogenous synthesis might actually be linked to fetal growth, specifically fetal length and head circumference. And the new data that's come out, which is a new aspect that we hadn't considered before, was shifts in creatine delivery to the fetus during different types of labour, with those being the most active or energy consuming, leading to higher creatine levels being delivered to the fetus. And then obviously, while dietary creatine levels were affecting maternal creatine levels, so there was a 30% reduction in creatine in our non-meat eating participants compared to matched meat eaters. This was not linked to adverse outcomes, but considering those shifts in creatine delivery to the fetus and in the placenta during placental compromise, this difference is something that we feel might need to be highlighted for women who choose to not consume animal products, as they might want to consider taking creatine supplements during pregnancy. And on that note, we obviously have new and exciting data about the benefits of creatine supplements during pregnancy for perinatal brain injury, or preventing that following acute hypoxia, coming out of our sheep studies. And those really being highlighted through a reduced accumulation of reactive oxygen species in the fetal brain after hypoxic insult in the creatine supplemented animals. And we will thus be looking at whether there are also connections between creatine levels and the levels of creatine in the brain of preterm infants, because this is a new area to look at, considering those new findings. But obviously, we still think that the potential use of maternal dietary creatine supplementation during pregnancy as a prophylactic treatment for fetal hypoxia and to prevent perinatal brain injury is really exciting. And as we move towards potential clinical trials in this space, we have recently conducted a consumer Prospective study, if anyone is interested in that aspect of the use of creatine supplements. So what pregnant women and their healthcare providers actually think about the idea of introducing creatine during pregnancy. And we're currently undertaking a pharmacokinetics and pharmacodynamics study of creatine supplements during pregnancy, so that if we do go to trial, we will so with an adequate dose of creatine. So just to finalize, is creatine a conditionally essential metabolite for successful reproduction? That was the question posed at the beginning. More evidence is still needed, but we are continuing to build strong evidence for the importance of creatine during pregnancy and the potential benefits of supplementation during pregnancy. So thank you very much. I look forward to answering any of your questions during the panel session, and a huge shout out to our team across Melbourne, Australia, also New Zealand, and in the US. It's a huge team, and we couldn't get this work done without everyone's involvement. So thank you very much for your time.